Hi, Colette. Thanks so much for joining me today on concentrating on chromatography. Could you walk us through your journey to becoming a senior director in biologics business at Waters Corporation?
So I joined Waters about 16 years ago. I have always loved science, specifically chemistry. So immediately following my PhD from Dartmouth College, I was looking around of like where I could get broad exposure because I wasn't sure what I wanted to do next. I knew academics was not a career for me. So I started to talk to different people. And one of the people that I spoke with quite a bit during my graduate career was the application support scientist at an instrument company. And I thought it was pretty cool, all of the different applications that she was able to see, the different kind of chemistry, the science, and the problems, and the exposure then she had to academic labs, to the federal agencies, the regulatory bodies, and as well as farm and biopharmaceutical companies. And I thought that sounds pretty cool. I want to do that because it'll also give me a little bit of a breath to understand what I want to do later on. Now, ultimately, in that time, working in applications, I found that I really wanted then to drive what we could do next in terms of what kind of insights that we could see, in particular for our technology, or the technology that we didn't have. That was really more of what it was of, I really want to measure this other thing over there. Can we just make some tweaks either to the software or to the hardware? Ultimately, that then led my pathway to, all right, we might not have the expertise in house. Can we buy that technology? So within Waters, I moved over to mergers and acquisitions and was able to be part of a few successful mergers and acquisitions and integrations, which ultimately led to where I'm at today, being a senior director for a portfolio of some technology that we acquired, light scattering technology, as well as particle analysis. And the fun thing about it is that I'm still very deep into the science because that's so important to know like where the science is going for what we should be designing next. But then it's also given me a bit of a view on the business aspect.
And what originally drew you to analytical science and ultimately into QA/QC for pharma and biopharma?
Yeah, so with our technology, definitely grounded in number one, analytical science. And then that then led me to QA/QC, both working in that area, assisting with people that directly do those kinds of jobs in farm and biopharm, assisting with their SOPs. And why I liked that area and I continue to like it is because I really enjoy certainty. There's so much chaos going around outside that the certainty that you can get from analytical studies really helps to ground me. And I think though the other thing is the competency. I mean, if you're getting all the way to QA/QC in that area, that this is the last final step for some of these groundbreaking therapies, to really make sure that they are safe and efficacious. So kind of that competency is a huge enabler for the success, but also to reduce on my side any kind of anxiety that I might have. You know, have all of these tests been done? Did everything pass? And there's that extension of like into my own life as well, like having the experiments run or actually trying things out themselves or trying things out. Some people might think that that is kind of anti to somebody that's very conservative, but it's really, I want to go and experiment and try it so then I can know does it work or doesn't work. One of the other things though, think about like QA/QC, some people have this conception because I work very closely with a lot of people in the QC department here in the instrumentation business side as well as in farm and biofarm. And they sometimes think that it's just like a checkbox, but definitely not at all. If we think about what they're doing in there, they're running the actual tests and really understanding what those molecules are, or really understanding is that instrument ready to be released.
And as you've gained this experience, how has your perspective on quality changed as biologics have become more dominant versus small molecule?
So while I was in grad school, I thought I was fortunate that we contracted out our services for a small mall pharmaceutical company where we were doing QC tests. And what I learned from there, as well as working with other pharmaceutical manufacturers, is that the small molecule side, it's there's a lot of certainty around the molecule itself. You know what the structure should be, you know what the chirality should be. There's instrumentation, there's tests that you can run to have absolute certainty on what that particular molecule is, as well as the excipients that might be compounded into it for eventual delivery. Now, with biologics, this is extremely different. So I've been fortunate also to move into that biopharmaceutical area, and extremely exciting area, extremely dynamic. And really in here, in the conversations, in the work that I've done in the lab, the people that I've spoken with, going to various meetings, for example, the nimble meetings that they have, the quality is about managing variability. If we think about biologics, probably a lot of people listening, they work on biologics, they're not static molecules. Like we might take snapshots with X-ray crystallography, but really they're dynamic molecules. And because they're so dynamic, and because the actual manufacture or development of those biologics are intrinsically linked to the conditions in which they're developed, what we have is a situation that they release on the process. So the product is the process. As long as we follow this particular process, that we have a particular temperature, we have a particular stir rate in the bioreactor, that we have this specific nutrients that are added at a certain time, then it's expected that that biologic is the same in that batch, batch B, as it was in batch A. But really, this is where a huge opportunity, especially in terms of QC, to change that around, because we have all of this instrumentation, we have a lot of depth of knowledge of years and years of monoclonal antibodies, right? We know these molecules too. So there's kind of this revolution that instead of thinking about the process being the product, that the product is the product, just like small molecules. I can know the glycosylation. I can know the extent of glycosylation. I can know the molecular weight with absolute certainty. I can know the higher order structure. So all of these things can be known. So even if they are very dynamic, it really should be like very empowering for that QC lab that they'll be able to report on all of these and have absolute confidence in what they're releasing.
Yeah, that makes a lot of sense. For listeners less familiar with these topics in general, how is QA/QC in biologics fundamentally different from traditional small molecule pharma?
Yeah, kind of like jumping off of the last question that you asked, biologics are heterogeneous by nature. So you might be doing single molecule measurements. Well, that single molecule is a little bit different than the molecule sitting right next to it in solution. And so what you're looking at is a dispersion, or well, it's polydispersity. It's actually, and sometimes people will report a polydispersity index. So you're looking at this population, and you're measuring the populations of molecules, and you have to think about, does that population fit into the overall model that we're trying to fit things to? So do they have similar enough glycosylation patterns or aggregation? So some might be aggregated, and another population or partial population might not. So that's one of the things, like when thinking about the biologics different from small molecules, is the heterogeneous population is just inherent with biologics. And really then it lends itself to very strong statistics required then to understand, is this different or not different? And it kind of extends out into the area of biosimilars of, is this a biosimilar? Is it the same? Is the sameness enough or not?
And as you could guess on the podcast called Concentrating on Chromatography, I've spent a lot of time talking about LC and high res mass spec, but I'd love to hear about how these critical analytical techniques are really supporting QA/QC today.
Yeah, so the nice thing is that we have a excellent view of LC or liquid chromatography used in that QC lab because of the type of supplier that we are. And then the other thing is actually, if you look at LC is foundational. It addresses at least 90, well, I'd say 80 to 90% of the attributes that are required in biosimilars. So the extension of that would also be in those innovator therapeutics. And LC is anywhere from SEC, so looking at aggregation, any kind of molecular weight, to ion exchange, to reverse phase. Thinking about, like you mentioned, high-res mass spec, so reverse phase before a high-res mass spec instrument. And what's really cool, what we've seen even more of, or even more so, for these bispecifics as well as some ADCs, So the antibody drug conjugates that you see more and more 2D LC kind of make its way downstream into those QC labs. As people are thinking about, okay, this molecule is hydrophobic as well as hydrophilic. How can we get it through efficiently? And one way is 2D LC of doing very efficient separations that way to get additional information. The other nice thing though is LC is a well-established technique. All the way back since insulin was first FDA approved, you'd see LC in those particular submissions. So the fact that it does have so much information with it, that it's just a really well-known, well-characterized technique, it continues to deliver for the biologics for critical quality attributes or CQAs. So there's the LC side. Definitely needed, addresses the multiple critical quality attributes. Now with the biologics, as they're becoming, as you're seeing more and more of these Frankenstein molecules or complex molecules, you're seeing more and more advanced technology too, like the high-res mass spec. So critical for identity, especially for post-translational modifications, whether it be a LIGO conjugated to a particular protein or a drug conjugated to a protein, and even extending out to radioligands, right? That's an extension of these kind of like these protein conjugates, as the proteins, the linkers, the cage, and then the radioisotope inside. And that's where absolutely mass spec is required. And then the other instrument that you start to see even more so is MALS, or multi-angle light scattering. Especially for these larger species like the lipid nanoparticles or virus-like particles or even conjugated vaccines. Because it gives you an orthogonal measure of molecular weight. It gives you additional information to the high molecular weight species as well of, is it an aggregate or is it an oligo? And that's kind of a cool thing of like pairing these different pieces together to really get this full complex picture of, I see something under this, or I see a shoulder on my chromatographic peak. And that's where you come in with the additional, like you said, the higher res technologies like mass spec, like light scattering to understand like what's actually sitting under that shoulder. So LC raises the flag, and then a detector downstream gives you even more information. In addition to that, though, I mean, we could go on and on about all the different techniques. I really love instrumentation, whether it be even things outside of the portfolio that I know, but instrumentation that I've used a lot. So capillary electrophoresis, to look at the charge variance. Aggregates, we spoke a little bit about that. Definitely you can measure it by light scattering with a UV detector, a MALS detector. And then alternatively, if you think about the more and more these larger species aggregating out, or these subvisible particles and how those are measured, whether it be by, if they're a little bit smaller, dynamic light scattering, or even some type of background membrane imaging or microfluidic imaging. So kind of a cool thing, like especially if you think about all of these are sitting in that QC lab, all the different instrumentation that they get to play with. It's pretty fun.
I was super excited to hear you mention 2DLC just because
Okay.
on the podcast we've talked about 2DGC a little bit. So I knew I knew it was out there somewhere with the LC side of things. So it's very interesting to hear about, and certainly worth a dedicated episode, I would say, to get into.
Definitely, especially if you start to talk to people that work on a lot of conjugated species, they're looking at 2DLC and then the bispecifics or multispecifics. 2DLC is the way to go if you think about the complexity there. And right, you've got all of these different antigen binding sites. You can do an affinity chromatography and then follow it up by other different separation techniques like an SEC or something.
And earlier you had noted that biologics are inherently heterogeneous. How does that complexity show up in QC testing?
So I did speak a little bit about the shoulder before.
Right.
And if you think about like either it shows up as a shoulder, so you think about chromatogram and you've got a shoulder and you ask yourself, did I just do a poor job separating it, or is there something actually sitting under there? And the other one is the heterogeneity is just looking at the full width at half max, or just like looking at the width of the peak, because then you know that you have increased polydispersity or in your particular sample. Now that'll raise the flag, but now you need to look under the peak, because you could easily, even a symmetric, like eluting species that elutes with, a pretty sharp peak, you could also have co-elution under there too. And so LC sometimes will raise the flag, and then you'll see like there's an issue, either whether it be any kind of like structural change that would cause it to elute at a different time, But then the other one, like the tricky piece, is actually talking to a few of our customers, and they're talking about co-elution of species. So you might have something that has like a contaminant with a similar molecular weight that then on an SEC column will elute at the same time. So those are particularly tricky to try to parse out and understand what's going on there. But then you'll also have co-eluting species where they'll have different molecular weights, but they'll have similar higher order structures. So if you think about an SEC column and what you're doing for calibration, you're basically looking at, you've got your protein ladder, and then you assume then that everything's globular and then it's going to elute at specific times. But we know not everything's globular. So we've also had basically times where people have come to us with their, especially with their light scattering instrumentation, and they said, yeah, it really saved my butt that time. And those are exactly the words that they use, because we had a contaminating species, totally different molecular weight. It was like this partially unfolded piece, but it so happened to elute at the same time as my molecule. And it really saved us in there that we could find it and then actually then change our purification method.
Can you give examples of CQA's critical quality attributes that are particularly challenging to measure?
Yeah, so it goes back to, I was just talking a little bit about the protein ladders and pieces. So in particular, one of the more challenging pieces to measure are the high molecular weight species. So many times that we'll look at chromatograms and people will see like, here's the monomer, here's the dimer, and here's all this other little stuff that's sitting out before those particular peaks. And we just call it high molecular weight species. But for a QC lab, that's not good enough. Okay, so they have to know what percentage of it. Great, you can get that through LC optical. But then it actually becomes even more critical, especially if you think about some of the hot molecules out there today, like the GLP-1As. It's not just good enough to say there's high molecular weight species sitting out there. They have to know, is that an oligomer or is it an actual aggregate that's sitting way out there? Because these form like super high molecular weight species. So not just dimers, it's even higher than that. And they need to be able to quantify as well as identify those differently from any kind of aggregates. So that's been particularly tricky. But the way that they do this now is through light scattering, because you can actually get the molecular weight of everything that's like sitting under that particular peak. And then you can know, is this an oligomer or is it just this huge aggregate sitting out there?
And this next question gets me excited for ASMS in about a month from now. But what recent advances in analytical instrumentation are having the biggest impact on QA/QC?
Yeah, so one of the things that we have seen a lot more is, I mean, you brought up ASMS, so I definitely have to bring up mass spec because we see it all the time, right? I go and visit customers, I visit their QC labs, and many times you'll see a very simple single quad instrument. So not intact, they're just looking at the peptides to just do confirmation. But now that these molecules are becoming even more complex, we're seeing more and more high-res mass spec instrumentation sitting in there because people are like, okay, peptide mapping is good, but what does it look like in its entire species? I mean, we've been speaking a lot about proteins, but like you can also extend this out to oligonucleotides of that diastereomer. If I don't have high-res mass spec, how can I tell whether or not like there is that diastereomer and what particular percentage is sitting out there? So more and more high-res mass spec connected on, typically reverse phase separation with the LC. And the other one is the multi-angle light scattering. So some of the molecules that have been really hot that require this kinds of in-depth analysis also includes lipid nanoparticles. So you'll see those with like the in vivo CAR T, as well as mRNA delivery, and even some siRNA and some other small molecules are delivered via lipid nanoparticles. And then for those, we're looking at not only what is the size, but also the encapsulation, which kind of brings us to other technology that's super exciting. If you are attending ASMS or you have in the past, is charge detection mass spectrometry. So no LC currently on the front end of that instrument, but it's pretty amazing what it can do for encapsulation as well as encapsidation for the AAV molecules. That you just need to know, is it empty or full? Or, for example, LNPs, is that mRNA partially sticking outside of the LNP or is it fully encapsulated? And so that's some of the initial work that the team's been doing in the application lab in Massachusetts.
That's great to hear. Yep, I will be there representing Organimation. My company will have a booth again. I think it's probably the third or fourth year in a row that I've done ASMS, and our nitrogen blowdown evaporators. The vast majority of our usage is concentrating samples after extraction, but ahead of LCMS, GCMS. So ASMS is a really great fit because unlike an event like ACS where there's 20 different types of chemistry, everybody at ASMS is focused on some area of mass spec. So it's a really great place to
Yeah.
be.
Yeah, it's a good community, definitely. I know the team here, like they've been working on getting ASMS ready for several months now. It's a big event for us.
And outside of AI, the other big topic that everyone loves talking about is automation. What role does automation play in modern QC labs, and where is it still underutilized, in your opinion?
Yeah. So it's interesting. Some people think about QC labs and they think, gosh, automation maybe not totally required there because there's not as many samples that go to QC versus something like discovery. But then there's the opposite that I've heard as well of we want to reduce that kind of variability that you could have from someone, for example, pipetting or diluting samples or adding different additives, you know. So that's one of the things like it heard it back and forth. But one of the trends that I have been hearing from quite a few people, and actually even today I had a conversation with one of my colleagues, we were talking about lights out labs. So the idea of this lights out lab of if we want to eventually get to that point, or whether it be lights out lab or real time release, really you're going to see more and more automation. But I think where it's underutilized is on the software side. There are many software packages that offer automation and analysis. And then in addition to that, they'll throw up flags if you're outside of particular specs, or they'll offer automation in for quality tests. If you're going to be running in QC automatically before it runs it, it does a quality check on the system to say, yes, it's ready to go. But outside of it, I think on automation and any kind of machine learning that we could also incorporate it into that, if we think about like the bigger picture, is the analysis that could be done on these larger data sets or these larger pools of information that we have. Right, we were talking a lot about all of these different orthogonal techniques where kind of their data sits in different software systems. And I think there's a really opportunity there to bring those kinds of like all of that data together in an automated fashion and really help to monitor the molecule. But then outside of that, it's also monitoring the environment in which that particular like measurement was done. Because that's one of the other things I attended an FDA seminar. We were talking about variability in measurement, and a few of the different innovators there, they were saying, or the biopharmaceutical companies, they were saying, actually one of the biggest challenges that we have is the environment. So we might have systems that they pass all of these specs. We have the automation that then tells us, or like on the system, like automated checks of it's ready to run. But we found that the environmental factors also play a big role into the data that we're collecting. So kind of bringing that kind of automated piece of in as well would be interesting on the environmental monitoring. And I think that like when talking about automation there, it'd probably be more like a machine learning application.
Yeah, I appreciate you focusing on both the hardware and the software side because you really need both of those components. What are the biggest challenges when transferring methods from the development into QC?
Method development. Yeah. I mean, like transferring the methods, it's making sure that that method is ready for QC. So not all methods are built for a QC environment. And so that was like, that's one of the biggest, the pieces of like the barriers is developing a method with that QC mindset. So the quality by design, right? And then that's why they have a particular name for it is that quality by design early on in that area. It's a huge burden, like developing the methods and then testing the methods, qualifying the methods. So it's easily a 6 month process to think about that particular transfer of something that's being done in development into the QC for the method side. It's a huge burden.
Do you have any general advice on how teams could better design methods early on to avoid some of those downstream QC issues that you've seen?
Yeah, so one of the things like that unfortunately they can't anticipate is sometimes there will be like changes to regulations. So put that aside, but usually people know it's not usually just sprung on them. So thinking about thinking ahead. And so thinking ahead, there's again another term for this in the regulator's playbook, and that's method life cycle management. And so this is method life cycle management as well as quality by design. And I mean, these are key phrases and in these playbooks because it means that you need to make sure that they stress the method early, like what could be happening in that later QC environments that we need to make sure that we account for earlier on in that dev space. And that will really help. Like, what are the variables that need to be considered in terms of a pass fail spec? And then really here on the method lifecycle management and quality by design, just having a software that enables that and kind of has that at the forefront of the mind is really important and can make a huge difference to minimize the time. So that software also, that particular software can use it in development as well as QC. I mean, that's the ideal mix. So you're not introducing new software as well and variabilities.
If you were running a QC lab today, what would be your top three priorities?
I mean, for any QC lab, it's data integrity. Like number one, data integrity. So whether or not it passes or fails, like there has to be no question of the data has been manipulated or not. So that's like number one. Number two would be reproducibility. So how do we make sure that we have reproducible results from instrument to instrument? So ensuring those pieces from tester to tester and from site to site, especially as you're seeing more and more of these labs and these manufacturing kind of coming back to the US with some different environmental impacts going on, especially with like the tariffs. So reproducibility is important, thinking about all the different factors that go into there. And then I think it's really like thinking about like those two things. It's the also making sure that we're training the scientists properly, like giving appropriate training. But I think that the third one would be education of those particular scientists, especially for QC labs. Sometimes people they think that it might not be as exciting as a discovery lab, but I totally disagree. It's the well, one, the power that they have in QC is amazing, and they should because they are the last step between that particular therapy and the patient. And so I think like that would be very important of a top priority for those people working in the QC lab of really emphasizing how critical that particular role is in terms of the patient, but also in terms of like them being the custodian of that particular data and understanding really what that data means. It's not a check mark of a yes, no, but understanding what does that shoulder mean, and then just really instilling that kind of curiosity in the QC lab.
I think just based on this conversation and the passion you have coming through in your answers and explanations, I think you definitely ended up in the right place in terms of where you focused on QA/QC. What's one investment, I should say, that delivers the biggest ROI, whether it be instrumentation, software, or something else?
Standardized workflows. And I'm coming from a specific, like you said, I'm coming from that kind of QC mindset or eye, but standardized workflows. It will just save time in terms of any kind of variability that it removes the variability if we have the standardized workflows.
Is there one general misconception that people might have about QA/QC in biopharma?
I think many people think that it is a very reactionary position where they're just measuring something, they've got their spec sheet. But QA/QC or QC, that QC environment, is very proactive. Think about all of the things that have to be done before the sample even gets to them. they're really thinking early on, even before the sample's there, of all the different variabilities, of all the different variables and variability that could be out there. So it's a very proactive environment actually before that sample comes in, of all the different steps and all the different pieces that need to be in place.
Do you have any general advice for labs working to build and the managers possibly leading high performing analytical teams?
Yeah, I think some of the better performing teams that I've been a part of or that I've even led have been those that very curious about what that particular ripple in the baseline could be. And then with that curiosity, they need to be then given room to grow to and time to investigate these particular things. And I think really a failure point here for people that, or for groups that are not high performing, is they're not given that little bit of leeway to investigate themselves and to go down that path and to be given that particular time. So I think that's very important for high producing teams, is to have that curiosity and then to be okay with failure, to be okay that we should fail. The sample shouldn't fail, but we should fail. We should try out new techniques and we could say, wow, this does not work. Or this is great. It's going to be so efficient now in my time because of this new particular tool that I'm using. So curiosity and the time.
And as I mentioned, the core audience for the podcast is early career chemists. What skills are most important for the next generation of QC scientists?
I'd say there's always the foundational piece, and that needs to be analytical fundamentals, understanding statistics, understanding an LC and how it works and being able to use that. But I would argue that data literacy is even more important now. I mean, if we are thinking ultimately to more and more automation, it's really understanding. You need somebody on the back end of it to understand that data, to understand the mass spec, and should a flag be raised or not? And I'd say the next generation needs to be more comfortable with understanding complex data sets, bringing multiple different instrumentation insights together to really have a complete understanding.
And let's say that there is a QC team that's curious about adopting some more advanced techniques, but they're hesitant. What question should they ask first scientifically rather than commercially, let's say?
Yeah, it's what decision do they struggle with today? And what decision do they struggle with their data today to either can they make that decision more confidently with additional analytical technology? So it's not about like, isn't this great? I've got this high res mass spec sitting out here. Now I can get to this very low ppb level. But is that helping them to enable to make more confident decisions to reduce the risk? Right? If you already have a method that gives clear information, reproducible answers, but if you find that you're relying on assumptions with the particular technology, then I'd say that's the time then to bring in orthogonal technology to have even more confidence in what's being reported out.
And I always like to put people on the spot regarding where they see their work evolving. What do you think QC will expect from analytical methods 5 years from now that it doesn't demand today?
Yeah, I mean, we already see it with a lot of the trends going on. I think it was like 2 weeks ago, the FDA was releasing some of their recent information on real-time evaluation. They've got a few pilot studies out of real-time evaluation of the safety of the safety results from a few different companies. So if we're going to that real-time piece that way, and if we ultimately want to go to real time, I really think like the big enabler here for anything to make these rapid decisions, it's got to be machine learning in that very deep data lake that so many people have. And then ultimately, like that will help to make decisions more rapidly, but as well as like more informed decisions. So it's pretty exciting that way, that like how the machine learning will enable those particular pieces. Now, in addition, I'd say that there's a big call also for machine learning and those very deep data lakes because these molecules are becoming so much more complex, right? We spoke a little bit about the bispecifics, there's trispecifics. So you really have to come out these molecules with so many different instrumentation, so many different data sets to have absolute confidence. And I think here, like centralizing the data, going deep into the data and understanding would be like a huge enabler of thinking out 5 years from now of will we be in closer to that particular real-time release to us being able to grab all of those data sets together, collate that information, and then automatically sync it up with the particular regulators.
In regarding the advanced techniques you had noted previously, was there one misconception that you'd most like to clear up about using those in QC?
Yeah, I think one of the most frustrating things that I hear is, well, we use that in development, and now we're just inferring the results that we see down here on this other instrument. So we don't need light scattering in QC, or we don't need mass spec in QC, because we just infer that when my chromatogram with optical detection looks like this particular profile that I got used to seeing all the time, that those other advanced techniques aren't needed. And many times that they're disregarded, not for it's going to take a long time to transfer them in, but they're seen as they're too complex to use. And that's tough for two pieces. Like one is they were used in development because they're offering critical insight into the particular molecule. I think that there's value to have them in that area, in that QC space. So now it's the next argument would be, okay, are these easy enough to use, or is there a high barrier to getting them into that QC lab? And for that, I'd say the complexity is not so much in the hardware. It's more of the software and the interpretation. And so that can be learned. And I think like I would challenge myself and other instrument manufacturers of if there are barriers, come back to us, let us know, and we can bring those down because that data should be downstream with the molecule as well.
Well, Colette, this has been super insightful. I really appreciate your time today and learning about all the great developments that are going on at Waters in the QA QC area. So thank you so much.
Thanks for having me on.